Hello and welcome back. Have you ever stopped to wonder why a stag has this magnificent rack of antlers, but a doe doesn't? Or why male peacocks are just, well, dazzlingly colorful compared to the peahens? It's not just some random fluke of nature. Nope, it's a code. It's a genetic blueprint that defines an organism's sex. And today, we're going to crack that code together. I mean, really think about it. From those antlers to the peacock's feathers, these things aren't just accidents. They're inherited traits, and they are major clues pointing us toward a really powerful genetic mechanism that's been working its magic for generations.
So here's our game plan. We're going to start with the big picture stuff, the differences you can actually see. And then we'll zoom way, way in, down to the microscopic world of chromosomes. We'll explore all the diverse and, frankly, kind of weird strategies nature uses to determine sex, uncover the strange rules of sex-linked inheritance, see how scientists proved it all, and then we'll put your new knowledge to the test.
Okay, let's jump right in with those visible differences. The official scientific term for what we've been talking about is sexual dimorphism. Sexual dimorphism is really just a fancy way of saying there are obvious physical differences between males and females beyond the reproductive organs. We see it everywhere, in human muscle mass, in a deer's antlers, a bird's bright feathers. The big takeaway here is that these differences are predictable, and that tells us they have to be rooted deep down in our genetics. So, if our genes are holding all the instructions, how do those instructions get delivered? Well, the answer lies in these incredible little vehicles of inheritance, the chromosomes. I want you to think of your DNA as a massive library of instruction manuals. Most of those manuals, 22 pairs in humans, are what we call autosomes. They've got the instructions for things like your height and your hair color. But there's one very special pair of manuals, the 23rd pair. These are the sex chromosomes, and they hold the critical instructions for determining sex. Now, you might be thinking there's just one way to determine sex, the human way, right? But nature is an absolute master of invention. Let's take a quick tour of some of the different systems it's cooked up over the eons. Just look at this table. The variety is incredible. In humans and fruit flies, we use the good old XY system. Males have one X and one Y chromosome, which makes them heterogametic. That just means their pair of sex chromosomes is different. But check out grasshoppers. For them, it's just about the number of X chromosomes. Males have one X, we call that XO, and females have two. And then, just to keep us on our toes, let's completely flip the script. In birds, it's the female who is heterogametic with ZW chromosomes, while the male is ZZ. And it gets even wilder. Fruit flies can also use a ratio of their X chromosomes to their autosomes. And in mosquitoes, it's not even a whole chromosome, but a single gene that calls the shots. It's just an amazing reminder of how flexible life and evolution can be. So all this diversity in sex determination has a really fascinating consequence. When a gene for something else, like eye color, happens to be located on a sex chromosome, it gets inherited right along with it. And that leads to some very unique and sometimes unexpected patterns. The classic example that helped us figure this out comes from fruit flies. So let's imagine we cross a male with white eyes with a true breeding female with red eyes. Now, true breeding just means she's guaranteed to pass on that red eye trait. And in fruit flies, we're going to assume the Y chromosome is basically empty when it comes to eye color. So all the important genetic action is happening on the X chromosome. Okay, now here's where it gets interesting. Let's do the reverse cross. A white-eyed female with a red-eyed male. The result is just fascinating. All the daughters get their red eyes from their dad, and all the sons get their white eyes from their mom. Do you see how the tree literally crisscrosses from one generation to the next? This was a massive clue that the gene for eye color had to be on the X chromosome. This pattern actually has a name, crisscross inheritance. So if you ever see a trait being passed from a father to all his daughters, and then from those daughters to about half her sons, it's a really good bet that the gene for that trait is located on the X chromosome. So seeing the pattern is one thing. But how did scientists get that final definitive proof? And maybe more importantly for us, how can we actually use this knowledge in the real world? This is where C.B. Bridges' absolutely brilliant experiment comes in. Let's walk through it. First, the cross. Based on what we just learned about crisscross inheritance, he expected all the female offspring to have red eyes. But he found a few rare females with vermilion eyes. Instead of just throwing out that weird result, he dug deeper. And this is the crucial part. He put them under a microscope, looked at their chromosomes and discovered they had an extra X chromosome. They were XXY. This was the smoking gun. The eye color was tied directly, physically, to the inheritance of the X chromosome. It was the proof everyone was looking for. Now, this is not just some obscure theory for a biology textbook. It has really important real-world applications, especially in agriculture. The process is called sexing, and it's all about being able to identify the sex of an animal at a super young age for economic reasons. Check this out. In the poultry industry, there's a gene for what's called barred plumage. It's like a striped feather pattern, and it's linked to the sex chromosomes. This lets breeders tell male and female chicks apart the day they hatch. How cool is that? It's the same idea with silkworms. It turns out male silkworms produce more silk, so breeders use a sex-linked color trait to identify and, well, get rid of the female eggs. It's all about maximizing efficiency in production.
All right, it is time to put your knowledge to the test. We've gone through the theory, we've seen the proof, and we've even looked at some real-world uses. Now let's see if you can apply what you've learned.
Okay, here's our problem. Hemophilia is a recessive X-linked trait. So what does that all mean? Recessive means you need the affected gene on all your X chromosomes to actually have the condition. And X-linked, as we now know, means the gene is on the X chromosome. We have a carrier female, meaning she has one normal gene and one hemophilia gene. She's perfectly healthy, but she carries the trait. So if she has children with a normal male, what are the chances that any son they have will be hemophilic? Let's use this little grid called a Punnett square to map it out. The mother's two possible X chromosomes are on the left, and the father's X and Y are up top. We only care about the sons right now, so let's look at the two boxes on the right. There's one outcome where a son gets the mother's normal X, making him a normal male. But there's another outcome where he gets her X that carries the hemophilia gene, making him a hemophilic male. So the answer is a 1 in 2, or a 50% chance, that any son she has will have the condition. And hey, if that was a little tricky, don't worry about it. We're going to have some more practice problems coming up soon to make sure this all really sinks in. So let's just do a quick recap. We started with the visible differences between sexes, which we now know is called sexual dimorphism. We learned that these differences are caused by a whole variety of genetic mechanisms, usually involving those special sex chromosomes. We saw how genes on those chromosomes can lead to really cool inheritance patterns, like crisscross inheritance. And finally, we saw how that knowledge was proven without a doubt and is still being used in very practical ways today. So I want to leave you with one final thought to chew on. We've seen that sex can be determined by whole chromosomes, by a single gene, by a ratio, and in some species, it can even be determined by the environment, like the temperature an egg is incubated at. So what does all that really tell us about the flexibility of life's most fundamental blueprints? Is there really one single set-in-stone plan, or is life constantly reinventing itself? Thanks so much for joining me today. See you next time.